Hello everyone, I am Mehrdad Bahadori. I'm doing postdoc at Brahms Laboratory at University of Montreal. Brahms Laboratory is the International Laboratory for Brain, Music and Sound Research. And now I would like to present my research, which is about transcutaneous vagus nerve stimulation and time perception. The vagus nerve, by being the longest cranial nerve, is a major component of parasympathetic nervous system. Vagus nerve extends from the brainstem and innervates multiple thoracic and abdominal organs. 20% of vagal nerve fibers are efferent, while 80% of them are found to be afferent fibers. Vagus nerve stimulation was first used to treat pharmacoresistant epilepsy by invasively stimulating vagus nerve over the neck using the carotid fork. In this figure, you can see what was the carotid fork. For the first time, the idea of non-invasively stimulating the vagus nerve was proposed by Ventureira in 2000. He proposed that the vagus nerve fibers can be stimulated by transcutaneously stimulating the vagus nerve fibers represented in external auditory canal using electrical stimulators. Interestingly, transcutaneously stimulating the vagus nerve is found to mainly activate the afferent fibers of the vagus nerve, but not the efferent ones. It is found that stimulating the afferent fibers of vagus nerve activates the locus coeruleus norepinephrine system via nucleus of solitary tract. And from there, the activations propagate to higher subcortical, such as hippocampus, and cortical, such as prefrontal cortex, insula, and motor cortex structures. These brain areas are found to play a key role in many cognitive functions such as working memory, response inhibition, and conflict monitoring. So far, several research has confirmed the improvement of these cognitive functions by stimulating the vagus nerve. Moreover, stimulating the vagus nerve is found to directly or indirectly influence the activities in multiple brain areas involved in time perception and processing, such as cerebellum, premotor cortex, and dorsolateral prefrontal cortex. To this regard, we hypothesized that the time perception might be improved, modulated by deploying transcutaneous vagus nerve stimulation. To test the hypothesis, we recruited 20 young adults. To assess time perception, we employed an Unizochronic detection task where participants were tasked with distinguishing regular tones from irregular ones. Additionally, they were asked to complete 3 questionnaires: the 9-scale PHQ, the 7-scale GAD, and the TVNS Adverse Effects Questionnaire. In the Unizochronic detection task, participants were presented with a sound containing 5 simple tones in each trial. They were required to determine whether the sound was regular or irregular. In irregular sound, all 5 tones had same inter-onset intervals, but in the irregular ones, the 4th tone was always delayed. In the time perception task, the shift of the 4th tone was starting from 180 milliseconds and was changing according to a staircase algorithm. If the irregular trial was detected correctly 2 consecutive times, the delay was becoming half. And if the irregular trial was just once being detected as regular, actually when the participants couldn't detect the delay in the 4th tone, the delay was being increased by 50%. I would like to mention that the regular tones were actually the catch trials being placed in the staircase algorithm, and the algorithm was being stopped after 12 reversals, which you can see them as turning points in this graph. For the stimulation, we have used the BioPac linear isolated stimulator. The study included 2 separate conditions based on the electrode placement setup. In the experimental condition, the electrodes were placed on Simba conchae, an area innervated by vagus nerve, and in the sham condition, the electrodes were placed on the earlobe, which is found to be free of vagus nerve. It should be mentioned that each session was carried out in a separate day with a 1-week interval. Here we can see the timeline of the experiment. And above, you can see the timeline of each session, which saying that the experiment was starting by filling the questionnaires, and after filling the questionnaires, the participants were being prepared for the experiment and the electrodes were being placed. Afterward, they were going through the explanation of the task and the training section. Afterward, the time perception task was being started, and at the end we have had the session to remove the electrodes and stuff like that. On bottom you can see the timeline within every trial, and in each trial we had 8 seconds, and the stimulation phase was 6 seconds, and we had within the 8 seconds we had the trials, the sound trials, and before the sound trials we had a preparatory tone. After the data collection, we have prepared the data for analysis. For the time perception task, for each experimental session, we have calculated the mean value of last 6 reversals for each participant, and we use that as irregularity detection threshold. Also, we have considered the scores for all the questionnaires that we have, and for the statistical analysis, in order to compare the sham and experimental condition within different participants, we have used the paired sample t-test, and also for the scores of the questionnaires, we have calculated the correlation between the questionnaire scores and the irregularity detection threshold, which was found by the staircase algorithm. The paired sample t-test revealed a significant reduction in irregularity detection threshold in TVNS compared to the sham condition. In other words, this result shows that the participants were able to detect smaller time changes as measured by the delay of the fourth tone in TVNS condition compared to the sham condition. I would like to also mention that the correlation analysis did not show any significant correlation between the irregularity detection threshold and any of the questionnaire results in either TVNS or sham condition. Now, the concluding remarks of this experiment. As the participants were able to recognize a smaller time shift in TVNS condition compared to the sham condition, this finding might suggest that the TVNS, which is the stimulation of the vagus nerve, can influence the cognitive functions related to the time processing. And also, one potential explanation for the underlying mechanism by which TVNS could influence the time processing can be the activation of locus coeruleus and norepinephrine system via the NTS, and thereby enhancing the functional connectivity of brain regions involved in time processing, such as dorsolateral prefrontal cortex and the other brain regions. And thank you so much for your attention.